There are reasons to believe that common simple repeated processes are fundamental to all information processing in the brain. Firstly, it is known that the entire neocortex is composed of one repeated, highly preserved unit called the cortical minicolumn. Secondly, it is known that the neocortex can process data independent of the modality by which it was acquired. A blind person can see with a camera that sends electrical inputs via the tongue. A deaf person can use auditory cortex to process visual information. This suggests that there are common processing tools between data modalities. It has recently been proposed that the function of cortical minicolumns is automatic association. This simply means using some data to match against a stored dataset to retrieve more information than was put in. An example is a phone book. We use a name and address to retrieve the name, address, and phone number. Automatic association is a simplistic data processing tool that can apply to any modality of information processing. This process can be animated. This box represents a dataset. As input data is matched, a threshold is reached at which the information is returned. Once activated, the dataset leads to an action represented by the circle. In addition to the cortical minicolumns, there is another simple, massively repeated unit that subserves the entire neocortex. Basal ganglia thalamocortical loops descend from the cortex and send a modulating signal back to the same area of the cortex. These loops are modality specific but have similar structure no matter what modality they subserve, suggesting they perform a common neuronal process. It's been suggested that this arrangement facilitates simultaneous or parallel data processing. Could the cortical minicolumn and the basal ganglia loop share a common purpose? How could automatic association be made faster? Automatic association is limited by the rate data is acquired, the rate data is matched, and the matching threshold. Contextual information could be used to modulate this threshold. If the match is more likely, the threshold could be reduced. For example, a round white object in a nest is more likely to be an egg than a golf ball. If the matches are more likely, the threshold can be reduced. If it is less likely, it could be increased. This process can be performed simultaneously as the data is being matched to datasets, meaning it can be performed in parallel. The role of such a threshold modulation can be illustrated in movement. Movement requires rapid analysis of vast amounts of data to smoothly contract muscle fibers with correct force and sequence. Consider this man wanting to move his arm from point A to point C like so. Suppose as the arm moves through point B, this dataset needs to activate the movement from point B to point C. Since the brain initiated the movement, it has the necessary data to predict what should happen. As data conserving the movement is analysed, simultaneous, parallel, context adjustment can occur, such that the action can be performed on time. Closely related datasets for similar movements would also exist. Let these be considered as these datasets and actions. Similarities between the movements would result in some data matching, but not activating the movement. This hypothesis may explain the motor symptoms of Parkinson's disease. Parkinsonian patients have underactivity of their nigrostriatal pathway. If this pathway was responsible for lowering the matching thresholds when diseased, these thresholds would be set too high. To use our previous example, at point B, the dataset is still not matched. The next sequence of movement can't take place, and the movement would stop until the matching is complete. 50 to 100% of Parkinsons develop dramatic nigrostriatal overactivity with medication, manifested as dyskinesias. If the nigrostriatal pathway is overactive, the matching thresholds will be too low. At point B, the wrong dataset would be loaded, leading to a series of corrective measures, which themselves would be flawed, leading to dyskinesias. If this hypothesis is correct, similar data processing errors should be presented in all data processing within the brain, because basal ganglia circuits are not just limited to movement. Specifically, when off, more data has to be processed to achieve the same outcome. When on, less data is used, leading to errors. Parkinson's disease represents a unique opportunity to test these theories. A subgroup of patients have dramatic changes in nigrostriatal activity within one dose cycle of their medication. The same patient can be tested on the same day, once when off and once when on, and dyskinetic, with a similar test. The only variable is the state of nigrostriatal activity. To test these predictions, I chose the task of recognizing a face in the crowd. This involves rapidly sorting large amounts of information. When off, patients searching for a face in the crowd should be less likely to find them, i.e. there is too much information to be processed. When on, patients should falsely recognize faces in the crowd, i.e. too little information is used and the wrong conclusion is reached. 16 patients with severe motor fluctuations were recruited with age-matched controls. Patients were not demented. Photos of crowds were taken around the Adelaide CBD. A computer program was designed which automated the test. Each test consisted of 40 trials. Each patient was tested on and off. Half were tested on first and half were tested off first. Age-matched controls were tested with a similar time interval between tests. In each trial, the patient was shown a face, such as this. When patients felt they were ready, they were instructed to press a button, which revealed the crowd. Half the time, the face was a match to someone in the crowd, such as here. If they saw the target, they pressed a button. But in the other half, the face was not a match. If they could not find the face, this was indicated to the examiner. No time limit was imposed. To analyze the results, I need to use signal detection theory. Signal detection theory was developed to quantify aspects of decision-making in recognition tasks. It was developed in the 1940s to understand radar operator skills in the war. It has since had a much wider role. A radar operator had to interpret a wide range of signals. Some signals looked more like the target than others. These signals can be arranged according to how much they resemble the target. This is called signal strength. There are two types of signal, those created by the target and those that are not, also termed noise. If these distributions are far apart, the observer may easily distinguish between them, and the task is easy. If these distributions are close, the observer cannot distinguish between them, and the task is very difficult. The distance between these two distributions is termed d' and is a measure of overall accuracy. At some point, the observer must decide whether the signal is a match to the target or whether it is not. This point of signal strength, at which the signal is reported as positive, is termed criterion. Everything to the right is reported positive, everything to the left is reported as negative. If the criterion is too high, much of the signal won't be identified, flashing, resulting in a failure of recognition. If the criteria is set too low, much of the noise will be reported as true signal, resulting in false recognition. These are the two indices I will use to discuss my results: d', a measure of accuracy, and criterion, a measure of response bias. The first results to discuss are the accuracy scores, d'. These are the scores for the BD patients on and off and age-matched controls. Parkinson's patients performed worse than controls. However, there was no difference between on and off states. These are the Parkinson's patients, these are the controls. This is consistent with other research that Parkinson's patients have recognition deficits, but the deficit is the same whether on or off. When we look at criterion, it seems that Parkinson's patients are making different kinds of mistakes depending whether they are on or off. When on, the criterion was too low, meaning they were likely to misidentify faces. When this seemed to shift around the normal criterion of controls. When off, the criterion was set too high, meaning they were less likely to find faces, and the controls fell somewhere in between. However, The power of this study comes from comparing the same patient results when on and off in a paired manner. Each line here represents the results of one patient on and off, and the controls. When on, the criterion is low. When off, the same patient in the same test on the same day has a higher criterion. There is no trend in overall accuracy. A hypothesis of the role of the basal ganglia data processing has been proposed. This led to an experiment which found novel results in Parkinsons patients. It does not mean that this model is true, as there may be other explanations. However, it does suggest that this model merits more investigation. The criterion shift cannot be attributed to less distraction when symptoms are in one or other state, because the overall accuracy is the same whether on or off. It seems likely that the nigrostriatal pathway, through whatever mechanism, is modulating criterion around a fixed or ideal point. Since the introduction of levodopa, a variety of researchers have found increased false responses on testing in Parkinsonian patients with dopamine stimulation, whilst overall accuracy on testing remains unchanged. Attempts have been made to attribute these to a frontal lobe deficit with dopamine. However, if this is the case, Parkinson's patients should perform worse globally on medication. Furthermore, a variety of non-frontal deficits have been reported in Parkinson's disease, such as in recall, in recognition, temporal sequencing, mental rotation, visuospatial impairment, etc. These deficits, when looked at individually, could be explained by a criterion model. To further test the model, the same data processing deficit should be present in all domains. I have some promising results from voice recognition tests. Stereotactic and proprioceptive tasks could also be designed. Animal models could be derived to test the predictions. Recently, there are neural networks available based on automatic association. This theory may be a blueprint to design such networks so that they can operate in parallel. Thank you for your time.